Hello everyone, myself Dr. JIBRAN Ahmed. Uh, I passed my MBBS from Maharashtra and I am currently pursuing MD in Pathology from Medical College Hospital, Calcutta. Now when I was a second year MBBS student, I wanted to read Robbins, and I even bought the book to read. I read it once, but the problem was I couldn't reproduce it. So uh, uh, I decided that I wanted to help all the second year students, fellow patho residents, NEET PG aspirants, dental student, as well as Ayush people who also have modern pathology in the syllabus, to make some videos which are short, concise and picture based with a number of flowcharts and which is aimed at the core concepts. So without wasting any further time, let us start. So first of all, I would start with a series of chapters from general pathology, starting from cell as a unit of health and disease till diseases of infancy and childhood. Today we start with cell as a unit of health and disease, and I've divided the chapter into four parts. So today I'm going to deal with the first part, the genome, histone organization, microRNAs, long non-coding RNAs, cell, plasma membrane. Now, the word pathology can be broken down into pathos. Pathos means suffering, logos means study. Now, the genome is made up of 3.2 billion DNA base pairs, and you will be surprised to know that 98.5% of the human genome is non-protein coding, whereas 1.5% of the genome is excuse me, is the protein coding genes. Now, 80% of the human genome is involved in a regulation of gene expression. Now, I'm sure all of you must have come across this term, epigenetics. Remember, epigenetics deals with the normal, okay? But if there is a dysregulation, it leads to malignancy. So epigenetics can be defined as a heritable change in gene expression which is not caused by alteration in DNA sequence. I would urge all of my listeners to have a notebook and a pen and to take down all the notes that I am giving. Let us first start with the non-coding DNA. So non-coding DNA comprises of five main parts. 1. The promoters and enhancers. 2. Binding sites that maintain a high order chromatin structure. 3. Non-coding regulatory RNAs which are comprising more than 60% of the genome. Again, this can be divided into microRNA, which is a hot topic, very important for NEET, and long non-coding RNAs. Fourth, we have mobile genetic elements, which comprise more than one-third of the genome, called as transposons or jumping genes. And we have fifth, the telomeres and the centromeres. Telomeres are the chromosomal ends, whereas centromeres are chromosomal tethers. A very important question. The most common DNA variation in human genome are the SNPs and CNVs. SNPs stand for single nucleotide polymorphism, and we will study about it in details when we read about genetics. And we have CNVs, that means copy number variation. Actually, what are SNPs? I will tell you in short. These are the normal variations, okay, that is present in a general population. Normal variations in the human genome in the general population. That is SNPs. And SNPs, an important part about SNPs is that only 1% of coding region is involved by SNPs, whereas the rest of them are in non-coding region. Whereas copy number variation, 50% involves the gene coding region, whereas the rest involves the non-coding region. Now, this is a beautiful image that I have taken from Robbins. First, I wanted to show you the heterochromatin. Heterochromatin is the dense inactive part. As you can see, this part is dense and this part is inactive, whereas the relatively lighter dispersed and active part is the euchromatin. You can see how the nucleus is arranged and how at the center of the nucleus we are having the nucleolus. Now, a part of this euchromatin when taken shows a chromosome. The short arm or the P arm, the long arm or the Q arm, the chromosomal ends called as the telomeres, whereas the chromosomal tethers, the centromeres. If we go into further detail, if we unwind these chromosomes, what do we see? We see these structures. What are these? These are actually histone cores around which the DNA is wound. And such an organization of a histone and DNA is called as a nucleosome. Multiple such nucleosome, when they are dispersed and active, they form euchromatin, whereas multiple such nucleosomes, when they are inactive and when they are densely packed, they form the heterochromatin. Now, these DNA on transcription give rise to pre-mRNA and mRNA, and finally on translation they form the protein. Now, what I want to say is the pre-mRNA is having the promoter region, having an enhancer region, and has what is known as exons and introns, whereas the mRNA doesn't have introns, because by a process called as splicing, the introns are removed. The mRNA has a 5' untranslated region and a 3' untranslated region and an open reading frame. Now, the nuclear DNA, as I have discussed, the heterochromatin is the dense inactive part, whereas the euchromatin is the dispersed active part. As we can see, these black things, these are the histone core, and DNA wound around the histone core forms a nucleosome. And when these nucleosomes are open, okay, when they are dispersed and linked by a DNA linker, they form the euchromatin, and when they are compact they form heterochromatin. As we have already shown, the pre-mRNA is having introns, which on splicing forms the mRNA. The mRNA has an open reading frame followed by translation and finally protein formation. Now, histone organization. These are the epigenetic factors, so these are present normally, okay, but whenever there is dysregulation it will lead to abnormalities or maybe cancer. Let us see number A, histone and histone modifying factors. Now, as we see, multiple nucleosomes are linked by a DNA linker to form a chromatin, and each nucleosome is made up of 147 DNA base pairs plus low molecular weight histone. HMF stands for histone modifying factors. These are the chromatin remodeling complex and the chromatin writer complex. The chromatin remodeling complex helps in exposing the gene regulatory elements like promoters, whereas the chromatin writer complex brings about methylation, acetylation, or phosphorylation. All these processes are known as marks. Okay, all these processes are known as mark, and a classical thing is that all these processes are reversible by means of chromatin erasers. So we read about histone and histone modifying factors. After that, we are having histone methylation of what? Of the residues lysine and arginine. Now, histone methylation can cause both activation or repression of DNA. Similarly, histone acetylation causes acetylation of lysine residues, and it can again cause activation by means of histone acetylase transferase or repression by means of histone deacetylase transferase. And we have histone phosphorylation of serine residues. It can again cause activation and repression of DNA. Now, DNA methylation brings about transcriptional silencing. And last modifying factor, as we see, is the chromatin organizing factors. All of these factors are the epigenetic factors. Excuse me. Now, the histone organization. A classic thing about histone organization is that we all saw that a histone is wound upon by DNA. This is one nucleosome. This is another nucleosome, which is linked by a DNA linker. There are four main types of histone. There is histone 1, histone 2, which is subdivided into histone 2A and 2B, and histone 3 and histone 4. Now, the DNA linker is formed by histone H1, whose main function is to stabilize the chromatin, whereas the nucleosomes are comprised of a histone octamer. Why we are saying octamer? Because we are having two each of H2A, H2B, H3 and H4. So this is a two-dimensional diagram, so we cannot appreciate the octamer, but in three-dimensional diagram we can appreciate the octamer. As we see, histone acetylation usually causes activation in most cases and leads to formation of euchromatin, whereas histone methylation leads to inactivation mostly and leads to formation of heterochromatin, whereas DNA methylation always leads to inactivation. Now we come to microRNAs. As we remember that microRNAs and long non-coding RNAs are actually part of the non-coding region of the DNA. So these are encoded by genes that are transcribed, but they are not translated. They belong to the family of non-coding RNAs. MicroRNAs do not encode proteins. They primarily modulate the translation of target mRNAs. It has a main role in post-transcriptional silencing of gene expression. That means that after transcription an mRNA is formed, but that mRNA is not allowed to express itself. There is silencing of that mRNA. These microRNAs are relatively short RNAs made of on average of 22 nucleotides. Has a role in normal developmental pathways as well as cancer. The human genome encodes around 1,000 microRNAs, and each microRNA regulates multiple protein coding genes. What microRNA does? They target an mRNA in post-translationally टारगेट mRNA को वो टारगेट करता है, which is in turn post-translationally suppressed. Each mRNA has a 3' untranslated region which determines its specificity for microRNA. That means that each microRNA is specific for an mRNA. Now this is a beautiful flowchart that I have prepared. Now, first, a microRNA gene encodes a primary microRNA, which undergoes processing in the nucleus to form a pre-miRNA. Now, these three steps takes place in the nucleus. The pre-microRNA, with the help of an export protein, enters the cytoplasm. Now this pre-miRNA is actually single-stranded, but it has a secondary hairpin loop structure because of which it becomes a double-stranded structure. This double-stranded pre-microRNA is trimmed by an enzyme called as a dicer. This produces a mature double-stranded microRNA which is 21 to 30 nucleotides in length. The microRNA unwinds and forms single-stranded microRNA. This single-stranded microRNA is incorporated into multi-protein RNA-induced silencing complex, RIS complex, and this RIS complex will attack the target mRNA. If the microRNA finds a perfect match, mRNA is cleaved and game is over there. If there is an imperfect match, although mRNA is not cleaved, but it leads to translational repression. So both these processes ultimately leads to gene silencing. Now, another important new concept is small interfering RNA. What is important about this small interfering RNA is that they are exactly behaving as microRNAs, but whereas microRNAs were endogenous, the small interfering RNAs are uh, uh, exogenous. They can be introduced in vitro into the cells. The synthetic siRNAs are used to study the gene function, being developed as a possible therapeutic agent to silence the pathologic genes such as oncogenes. Now, long non-coding RNA, very important for NEET. It regulates the gene expression, can cause gene activation, gene suppression, chromatin modification in the form of methylation or acetylation, or stabilize the primary or secondary protein structure. Very important is an example of long non-coding RNA. This long non-coding RNA plays an important role in physiological X chromosome inactivation. Now we see the cells. Now, very important from NEET point of view is you see the cytosol occupies 54% of the cell volume, okay, and the main function being metabolism, transport, protein translation. Mitochondria, maximum number of mitochondria present per cell. So there are 1700 mitochondria per cell. And we all know the main function being energy generation and apoptosis. The rough endoplasmic reticulum is associated with synthesis of membrane and secreted protein. The smooth endoplasmic reticulum deals with protein modification, sorting, and catabolism. Nucleus involved in cell regulation, proliferation, and DNA transcription. Endosomes involved in intracellular transport, export, ingestion of extracellular substance. Lysosomes are involved in cellular catabolism. And very important, the peroxisome, again an important exam question. An MCQ question is that they are involved in the metabolism of very long chain fatty acid. Now the role of smooth endoplasmic reticulum. Now the smooth endoplasmic reticulum are found in abundance in the liver, where they help in lipoprotein synthesis, and in the gonad help in steroid hormone synthesis. Proteasomes are grinders that chew protein. We will discuss about proteasomes in details in our next part. Cytoskeleton is involved in cell movement and maintenance of cell shape. Now, the last part of this lecture. Now, this is what? This is actually a plasma membrane. As we all know, a plasma membrane is a lipid bilayer. You can see that there is a head part and there is a tail part. This is the tail part, which is lipid. Now the head part is a hydrophilic or water loving part. This part is hydrophilic or water loving part, whereas the tail part is the hydrophobic part or water hating part. Now very important now are the lipids. We will first discuss the lipids that are present in the plasma membrane. On the outer side are two important lipids, phosphatidylcholine and we are having sphingomyelin. Whereas on the inner side, towards the cytoplasmic side, we have phosphatidylethanolamine and phosphatidylserine. Now, on the outer side, we see something called as the lipid raft. Now, we should understand one thing, that the lipid raft is, we should understand one thing, that the plasma membrane or is a bilayer, and it is a fluidic in nature. That means uh, it flows. It is not at all constant. It is fluid. But lipid rafts are a stable portion of the plasma membrane. They are the stable region. These parts are not fluid, but they remain stable. So these are the stable parts of the plasma membrane called as the lipid raft. On the outer side, we also have glycolipids. And on there are two more proteins, the phosphatidylinositol, which are present both on the inside as well as outside, and we have cholesterol, which are present again on inside and outside. Also, the plasma membrane is comprising of proteins, the most common of which are the transmembrane protein. Then we are having certain proteins towards the cytosolic end called as cytosolic protein, and certain proteins on the outer end called as GPI linked protein. And there are certain extracellular proteins and lipid link proteins. So again, the plasma membrane is a bilayer, the fluid mosaic model, and it is asymmetric. Phospholipids, the plasma membrane is comprised of phospholipids, proteins, and cholesterol. Now, the phospholipids on the outer side, as we have discussed, are phosphatidylcholine and glycolipids and sphingomyelin, whereas on the inner aspect we are having phosphatidylethanolamine and phosphatidylserine. Now, the function of glycolipids and sphingomyelin is they have a role in cell to cell or cell to matrix interaction. They also play a role in inflammatory cell recruitment and sperm egg interaction. Whereas phosphatidylserine is a negatively charged molecule, and if it comes out of the cell, it will trigger cell death in the form of apoptosis. And also, the phosphatidylserine is a cofactor for blood clotting. Now, as we have read, phosphatidylinositol is present both outside as well as inside, and this phosphatidylinositol gives rise to diacylglycerol and inositol triphosphate, which act as second messenger. Again, cholesterol is found on both surfaces. As I've already mentioned about lipid raft, these are stable, distinct lipid domains. One thing I wanted to mention is this is the lipid bilayer model, the fluid mosaic model. Look at the head, which is hydrophilic, and look at the tail, which is hydrophobic. Now, there are different proteins. There are different proteins which are also present in the cell membrane. Those are the integral or the transmembrane protein. They translate the mechanical force and chemical agents across the membrane. Then we have certain cytosolic proteins or lipid linked proteins. We have GPI anchors, which basically attaches the proteins to extracellular face of the membrane, and we have other peripheral membrane proteins. Now, the basic function of these proteins are ion and metabolic transport, receptor mediated uptake of macromolecules, cell to ligand and cell to matrix, and cell to cell interaction. Plasma membrane may function together as a large protein complex, either in the form of an aggregation of protein under the control of chaperones in rough endoplasmic reticulum. We will read about chaperones later. Chaperones are substances which have a role in protein folding. We will read about it in details later. And secondly, the plasma membrane may function together as a large protein complex by lateral diffusion in plasma membrane followed by complex formation in situ. Thank you so much. With this, I will end the first part of this chapter, and stay tuned for the second part later. Thank you so much, guys. And please don't forget to subscribe. If you like my videos, please subscribe and like my videos. And please comment if you want me to make any other changes. Thank you so much.